this lecture, you've got the self-study one that I'm not going to cover, and then you've got, because that one is because it's the MHC one, and from what I remember when I took the course, that basically was just going off of what I already knew from Immuno, and Dr. Blank brings up MHC quite a lot. It's in this lecture, not so much with the last couple ones he talks about it a little bit, and so that lecture that he gives you to do on your own is kind of a lot of stuff that he just wants you to synthesize information that you've already seen. And then we've got the personalized medicine one. So I'm going to do another review on Friday. I'll do Friday morning, Friday afternoon, and then that'll be that. So that Friday one, I'll go over personalized medicine, and then I'll be there for any questions you guys have. Since there's so few lectures, I don't know if it's worthwhile to do a review of all the lectures. So that way I'll just be there Friday, and then you guys have all the weekend to worry about Immuno or any of the exams you want to look at. So yeah, let's just get started into this one. So like I said, this is what I posted online. You're more than welcome to just use this, but there's a very good chance stuff's going to be on the exam that's not on here. Or I probably go into a lot of detail on stuff that Dr. Blank doesn't really care about. So I highly recommend you read the article. It's not that bad. And like I said, most of this stuff we've actually already seen. Like you guys already had to know this at one point. If you didn't know it before, that kind of sucks, but we have to know it again now. So really there isn't that much new information that we have to go over. It's just kind of bringing it all together for cancer genetics. And one thing to just keep in mind is that I don't know when this article was posted, but I think it's kind of old relatively. So like I guarantee like a lot of the stuff that, at least for the exam, know what's based on here, but in terms of like real world information, I'm not sure how up to date it is, and I'm sure there's a lot of new information available. But yeah, so we'll just sort of get started now. If you have any questions, let me know, and we'll sort of run through it. Just my disclaimer. So, Dr. Blank, you guys can't get mad at me if it's not in the exam. So yeah, so basically cancer, all of these things I think hopefully everyone has a pretty good or decent idea of. The one thing to keep in mind, I kind of been saying it over and over again, is that really no two cancers are the same, even if it's the same type of cancer, like pancreatic cancer in me or pancreatic cancer in you. That doesn't mean that they're the exact same sort of, they have the exact same sort of symptoms, they're not going to be treated the same way, different survival rates. All that stuff is different on a case-by-case basis, and that's because, by definition, cancer is due to mutations. So mutations are going to occur, and the net result of those is it's going to lead to unrestricted cell growth. So cells are going to grow very, very rapidly, but within there's a bunch of different possibilities that we could see. So obviously, they're going to want to divide. They're going to divide without the regulation. They usually are going to lose apoptosis. Oftentimes, they can spread to other areas of the body. That's metastasis. So that's literally just leaving one area of the body and then migrating somewhere else. One thing to be aware of is that some cancers, they only metastasize to only other certain regions. So the example I think they had talked about is prostate cancer. So prostate cancer obviously will start in your prostate. If it does metastasize, it's going to always go to the bone. Almost always, I believe. So different cancer types will have sort of different preferences when they do metastasize. And typically when we're talking about survival rates, when you get to metastasis, that's where it is a much more negative outcome for the patient. So if you end up going in too late for a checkup or for a growth or whatnot, and at that point it's metastasized, usually that's going to be mean a very, very poor outcome for you. What do we do to check for metastasis? Like what's typically, what will they look at? So if I see maybe you have breast cancer, what's my first thing I want to look at to see if it's metastasized? The lymph nodes, right? Because the lymph nodes, they're going to be draining that area, and if any cancer is going to metastasize, it usually will go through the lymph nodes to then migrate somewhere else. Because the lymph nodes are going to be collecting all basically the lymph and all the sort of like the debris in that area, then move somewhere else. Angiogenesis, we've already talked about. That's basically where blood vessels are going to basically grow towards that tumor. Tumor, once again, they're cells. They're abnormal, but they're still cells. And what do all cells need to survive? They need blood. They need oxygen and nutrients. So you have to have, typically if we get a lot of angiogenesis going on, that's going to lead, once again, to poor outcomes. Because now, basically, we could have this entire blood vessel rerouted or moving somewhere else to this tumor. Do you guys remember what the chemical is involved with this? VEGF. It's that vascular endothelial growth factor. Obviously, oftentimes they can have increased survival. They can evade the immune system by changing MHC, mutating things like that. But this is the biggest thing, is that not every cancer is going to have all of these characteristics. They can have some of these, but they usually don't have all of them. I believe the one that you have to have is unrestricted cell growth. So growing very rapidly, dividing very rapidly. But outside of that, all these things could be there or may not be there, or they could develop over time if the cancer is left untreated. And once again, like another example is here, brain tumors, they don't metastasize. Granted, they're inserting your brain, so that's not very good, but they're not going to metastasize. And lymphomas, those are basically blood diseases. So it's going to be things in your blood, like T-cell lymphomas, things like that. So technically, they don't metastasize, but they're everywhere throughout your blood. So example stuff like that. And once again, all cancers, it's due to mutations. The things are going to be, we're going to be changing the DNA one way or another, or we're going to be losing structures of DNA, or we're going to have more copies of one gene or lose copies of a gene. All those things are in the result going to lead to this unrestricted cell growth. And I believe, we're going to get there in a second. So yeah, different kinds of mutations we can talk about. We've got the effector proteins, and basically these are the ones that are going to be directly responsible to the cancer phenotype. So remember when we looked at like the last lecture on the TCGA, and Dr. Blank did studies where he looked at all the possible mutations and then compared those to the ones that mutated oncoproteins and how those were the big important ones. Those are the effector proteins. So in the case of, we're talking about cancer research and just information dealing with cancer cells, effector proteins, those are going to be the ones that are going to directly, when they're mutated, they lead to cancer. So that could be like changing retinoblastoma or any sort of thing that's going to either be responsible for apoptosis or cell cycle. Things like that, when they're mutated, that's when we're going to have the effect. And oftentimes, it's usually not just one mutation. It's not like the one thing happens and then we're screwed. It's usually one thing will happen, another thing will happen. It's this cascading effect. Over time, we get a bunch of mutations. Out of all those mutations, the changes to those effector proteins are going to be obviously the very big issues that we're going to see. Aside from that, we could also modulate regulatory proteins. So when you think of this, think of like transactivators. This is the idea that the actual gene, that the effector gene that's making the protein, let's say that's going to allow for apoptosis, that gene is still normal. I didn't mutate it, I didn't get rid of it, I didn't amplify it or get rid of its copy number. What did occur though is transactivators or other things that are going to bind to upstream of that to then allow it to get transcribed or not, that's what's going to be lost. Granted, in this case, it would need to be an inhibiting sequence-specific DNA binding protein, if my example is apoptosis, right? I want to have this protein available to go into apoptosis if other things are screwed up. If I then get rid of, or I somehow mutate it and I get way more inhibiting SSDBP, I'm now not allowing that apoptotic protein to get made. So now I can't go into apoptosis, then I'm going to have the issues. You can think about the flip side with like a tumor or like an oncogene with an inhibiting or an activating transactivator. So that's the general idea. So think of regulatory proteins, think of those transactivators, those SSDBPs that are not going to be changing that protein, that gene in particular, but going to be working upstream to then affect how much of that protein we're going to end up getting. And like I said, to keep in mind, since every cancer is unique in its own way, and it's all due to random mutations, we at this point now we don't know everything there is to know about any cancer really, let alone all of them. So all of this stuff it's cutting edge. It's where a lot of the research is. But sort of keep that in mind going forward that we don't really know everything, and there's going to be some times where we're not going to fully understand what the hell is going on. Maybe even in this PowerPoint, maybe some things that don't really always make sense, but that's just sort of how it is. So metastasis. Once again, this is that idea of a cancer. Basically, it'll start in one area due to mutations there, but it can actually metastasize and migrate to other areas of the body. And then it can basically what happens is it can pop off from where it is and then move around, and it has basically these sticky appendages with it where it can then attach somewhere else, and then now it's in another area of the body. That's the general idea. But we actually don't even know why metastasis occurs. So there are basically two options. The first option is through all these mutations, we end up basically having an entirely unique process that a cell wasn't able to do at first. So it's sort of like, it's basically, you know how when we talk about cancers, we're mutating and we're changing like the normal machinery of the cell? The first hypothesis is that this isn't any normal machinery, it's literally an entirely new thing that only occurred in these cancer cells. The other hypothesis that I'm more of a fan of is the idea that we're still using the normal machinery of the cell, the normal processes, but we've changed them slightly. So think about the idea of how, like, epithelial cells, by definition, like, what are epithelial cells? Have we talked about that in the anatomy yet? Probably not. Probably for histo. But epithelial cells, they're sticky. They always stick together. They're going to be in long layers, long strips, and they're always connected all together. They're different from connective tissue where it's more spread out. So one example could be the fact that, well, maybe when the cancer, in terms of how it can migrate and then stick to somewhere else in the body, maybe it's using other certain proteins that are important for epithelial cells, like some of those desmosomes and those cell structure, those cell plasma membrane things, to stick somewhere else. So in this hypothesis, it's still the normal function of those cells, but we've changed it slightly. Versus this hypothesis, it's an entirely new thing that we've never seen before in cells. This is sort of an idea of why we don't really know that much, because there's still so much that we haven't really discovered yet. So obviously, how do we acquire mutations? There's actually more than just two, but we're going to start here for the article, and at the very end, we're going to add another way that we can get basically loss of genes or gain of genes, things like that. Obviously, we've got the Mendelian inheritance. That's just sort of the idea that you have, two copies, one from mom, one from dad, big A, little a, however you want to think about it, and then you can lose one or lose two. And then you've got homozygous and recessive. But however, for a lot of these, when we're talking about them getting inherited, that means that when the sperm, where did the mutation occur? If we're talking about inherited, so like you have it from birth, right? So if you had it from birth, where did the mutation have to occur? In the germ cells from your mom and your dad. So the sperm was screwed up, or the egg was screwed up, or right at fertilization they got screwed up. And then that means every cell in your body is going to have that, right? Every cell in your body will have a good A from your mom, but a bad A from your dad. And if it's a homozygous recessive trait, we're still okay, but then what happens over the course of a lifetime is I can mutate this good A, and that's this second one. So that basically, as we go through successive mitotic divisions with your cells, you can lose that other one. And this is typically how a lot of cancers, a lot of genes in general are going to be functioning. It's not usually just one over the other. So once again, mutations are random. But going back to that idea is that if I have two really good A's, the probability of me losing both of these A's getting mutated is very, very, it still happens, but it's very, very low. As opposed to, I have one good A, already one bad A, I just have to mutate this one, and now I'm in trouble, all right? So really it's the combination of these two working together. And this is often why, for a lot of cases, in a lot of even cancer types, we see that there is a sort of a, if obviously a certain cancer line is running your family and there's a genetic component, then obviously you're going to be more at risk for that if you run, like what's the big example everyone talks about, that we're going to talk about, is breast cancer, right? Where there's a predisposition based on your family genetics, what you inherit from your parents, that can greatly affect sort of the probability of you also developing that cancer type. We have the idea of penetrance. This is from the other lecture, so make sure you understand this pretty well. The idea here is that the biggest example that I like to think of is that something can be dominant but have low penetrance, and the best example I think of if it's a multifactorial disease, so multiple genes affected. So yeah, one gene is screwed up, but what if there's like 25 genes in total that are going to affect this trait? So even though the one, it will be a dominant based on its definition by Mendelian genetics, it could still have low penetrance because it's not just the one bad gene that's going to cause it. It's this bad gene plus I need other bad genes also that are going to be mutated for that to occur. So yeah, keep these in mind. The third one on here that we're going to talk about is loss of heterozygosity. It's at the very end of the paper. It's not really a mutation by definition, but it's similar, so we'll touch on that at the end. Numbers to know: about 5 mutations in coding regions is typically what's going to make cancer fatal. Once again, this has to be in, I believe it's going to be genes that are going to be responsible for cell cycle, for apoptosis. Why is it never just one, really? And the answer to that is that, remember, the cell has a lot of fail-safes. The cell, we've evolved to the point where if we lose one, the cell could be okay, or at worst, it'll say, oh, I'm not okay, and just kill itself. So usually it's not just one thing that gets screwed up, and then the entire cell. Now, this is, like I said, I'm talking in general. There are some where there is basically one mutation. But overall, it's typically about 5 mutations in coding regions. That's what we're going to say that will make a cause of cancer to be fatal. So why do cancer cells kill you? Basically, all the things we talked about. Unrestricted cell growth, it's going to pull blood vessels away. You only have so much blood in your body. So if I have a tumor here that's taking all the blood, it can't go to any of my other organs. You're basically using so many nutrients, so many resources that over time, it can just sort of overpower you. So yeah, sort of keep that in mind. But yeah, that's the other thing. That's a number. I'm sure this number isn't really real now because this paper's pretty old, but for Dr. Blank, know it as about 5 from there. So there are different case studies. All of these we've already seen. You've already seen. So for example, they have the RRD 251. I wouldn't know it by that name. It's to treat melanoma. I basically would know it's for melanoma. This is what it is, and what it's going to do. It's going to prevent RAF kinase and RB. So this all goes back to that retinoblastoma pathway, or in particular the BRAF one. Remember the BRAF? We have like the RAF kinase, RAF kinase, all the random names, and they have examples of that. So if you do not remember this well, go back to the yellow highlights on exam 2, and at least go read over that again. It's like 4 or 5 slides. I posted in it's PowerPoint on Facebook. So make sure you're at least comfortable with that. You don't need to memorize that entire case study, but this BRAF pathway is going to come up, I think, 3 times in this lecture. I told you it would come back, and it's back, all right? So just make sure that you're at least comfortable and understand the general idea of how things are related in that pathway. And so what this drug is going to do in particular, what's the end result of that BRAF pathway? Do you guys remember? What's it going to change for gene? It's going to increase the genes and make proteins for what? For cell division. So it's going to make you divide, divide, and divide. So obviously if we mutate that and if we make it always active, we're going to have cells constantly dividing. So what this drug does, it screws up that pathway, and then what would be the natural result of that? It's going to facilitate apoptosis. Because we're basically halting the cell from going to the next phase. So what happens when that happens? That's like a check marker, right? You have those checkpoint proteins that normally they're going to go, if something's screwed up, they're going to be, nope, let's pause it here. If a cell is paused for too long, it is just going to die. It's going to go through apoptosis. That's what's going to naturally occur. So by having this drug and preventing this pathway, we're basically going to pause the cell up into a point that then the cell is going to start creating these pro-apoptotic proteins that then will cause that cell to die. That's what we want, because what are these cells? These are the cancer cells. They're dividing unrestricted cell growth. One thing to keep in mind, for a lot of these studies, we can use immortalized cell lines. I think, what was the example? There's like the HeLa cells, right? Where basically they took, from a patient, they took these cells and they were able to freeze them down and they continuously divide. So we have these cell lines. And what's great about this is that every cell, obviously aside from where the mutations are going to occur, is going to have the same DNA in all the cells in the body. So we have the patient's DNA, we can work with those, but really the really hard thing that we haven't really perfected yet at all is that not only do we need to have the cell line, but we need to make sure that it's the proper microenvironment. So that means that the cell in the body, I need to have every other chemical and protein and whatever else in the body, I need to have that in the Petri dish. Not everyone, but a lot of the big ones. That's very, very hard to do. That's where the big area of research is trying to do, is to basically try to figure out the correct way to create these tumor microenvironments. And once again, these microenvironments are different on a patient-by-patient basis. So it's not like we solve it once and then we can use it for all the different types. So it's really very difficult to do this. But this is where what we're trying to do, and in some cases, studies have accomplished for unique patients and unique scenarios. So yeah, so basically what I do on this, I put the picture with all the shit on it, and I talk about it on the ones after it. Because you don't need to know this entire thing. Did he highlight the stuff? He put like a few highlights, right, of like words or something? Yeah. Yeah, so like, again, me reading, I was like, I'm not just going to like assume that's what it's going to be, because I'm sure a lot will come from the yellow highlight stuff, but it's really just like key phrases, right? It's not even like whole sentences. So really just trying to know as best you can is ideal. Like I said, if you don't want to, so I try to go into as much detail as possible. So I'm going to break down basically this picture into a bunch of different sections. So we have cell cycle. Once again, everything we should already have a good idea of. You got G1, S, G2, and M for mitosis. Keep in mind, in between these, you're going to have those checkpoints, right? Those checkpoint proteins. Their goal is, I want to go through G1. What happens in G1? G1 is just growth, right? It's just going to be growing. Things are going to be going naturally. Near the end of G1, what do I need to have if I'm going into S phase? I need to have, so S is for DNA replication, right? So all those proteins in the first exam, the DNA polymerase, all the other things we have to have, all the histones for more DNA, these all need to be made around here. So before I go from G1 to S, you have checkpoint proteins, and their job is going to be, all right, do I have all my histones? Do I have all the proteins I need for replication? If yes, great, let's move on. If I don't, they're going to pause the cell here, and we're going to wait a bit and say, okay, let's make them the